welcome to my channel BioWorld once again. So today with the chapter inheritance and variation, that is genetics, I've come with a topic called incomplete dominance. In my previous video I talked about monohybrid cross, where we discussed about law of dominance and law of segregation by showing the crosses between the homozygous tall and recessive dwarf plant and producing their resulting their offspring in F1 and F2 generation. So what we found in our last video that a character, when it is crossed, there is a category of dominant and recessive. That one of the character which is dominant would be expressive and the other one would be suppressed in presence of dominating character. So every time we crossed, we found that whenever homozygous trait is crossed with homozygous recessive, dominating with recessive, always the dominating feature get expressed. Whereas in heterozygous also, the dominating feature would be expressed in phenotype, whereas genotype remains the same. That is, gametes would be different. Genes would be different. So what is gene? Gene, you know, that's a segment of DNA or the part of chromosome that carries the trait or a character. So always in a diploid organism, the pair of gene, that is a contrasting pair of alleles, we say, contrasting pair of gene, one either would be same to the other or it would be different to each other. That is, one would be dominating, the other would be recessive. So in this particular topic, incomplete dominance, there is little twist where we will not get the same result as we got in monohybrid cross in Mendelian experiment. What happens here, you can see, we'll discuss it with the diagram given on the board. Here we are going to see if F1 had a phenotype that did not resemble either of the two parents and was in between the two. So the offspring, the one which will be produced in the F1 generation, if it is not resembling either of the parent, that is, it is in between them, that is called incomplete dominance. The example is the inheritance of flower color in the dog flower, that is snapdragon or Antirum sp species, that is. So what we see here in parental generation, that is gametes in parental generation, we have taken red flower, capital R, that is homozygous, whereas another one is white flower, small r. They were crossed, and what was found in F1 generation, that according to Mendelian experiment monohybrid, it should be red, but it was same, that is hybrid genotype or heterozygous genotype, that is capital R and small r, but instead of getting red flower, the color of the flower was produced pink. That means it is neither resembling either of the parent, that is nor red flower or white flower. So it has come in between, that is pink. So this happened, why? Because the allele was unable to get expressed completely. That means the gene or allele was not completely dominating in nature. In the second generation, that is in F2, the cross was means between the offspring itself, that is self-crossed. So RR as a gamete, RR, that is capital R and small r. So what was produced? R with R, that is red, R with R, that is heterozygous or hybrid, these two, but they either of becoming red, they have turned out to be pink. And recessive will come together to produce the recessive trait, that is white flower. So please see the Punnett square here, how the crosses was done and what was the offspring produced. So what we got in F2, one red flower, that is completely dominating one, another two would be pink here, and the last would be white. So the ratio of genotype would be same as it was found in mental monohybrid cross, but what happened in phenotype, either getting red, the pink flower was produced, that is incomplete dominant feature of the trait. That means allele was not completely dominating. Hence, in between character was produced. So, what would be the phenotypic ratio? That is 1 red flower, 2 pink flower, 1 white. That is 1:2:1. This was phenotypic ratio. Whereas, what was genotypic ratio? Would be same, RR1, capital R, small r2, and RR1, that is 1:2:1. So slightly different from the Mendelian ratio in monohybrid cross, where the phenotypic ratio was 3:1. Here we are getting both phenotypic and genotypic ratio same, that is 1:2:1. So this was incomplete dominance, and very easy part to go through. Go through it. Next, with coming with the codominance of the cross. Let's see. Stay tuned. Keep watching. Thank you for watching.